in our last video, we talked about the 7 most common functional groups, which are clusters of molecules that endow, that they confer the bigger molecules special functions and structures and properties. So today we're going to mainly focus on 2 of them, which are carboxyl groups and amino groups. And these 2 collectively together, and they meet other atoms, and they collectively form amino acids. So there are 20 most common amino acids when we're talking about biology, and we'll need to group them into 4 groups, 4 subgroups, each comprises of several amino acids. And the group process is based on these unique properties or structures or charges of the amino acids. So today, we're going to talk about how that you can memorize the amino acid groups. I'm a lecturer today, and my name is Adrian. You may also follow me on my Instagram account. Now, this video is powered by Peter Tutor 2016, and all Peter Tutor videos are now available on both YouTube and Bilibili for your audience. And just to clarify, all my contents in this video and I think in my previous videos are all extracted from Campbell Biology 10th edition and written by Neil Campbell and Reese. Okay, without further ado, let us have a recap about the amino acids. So this is typically the structure of amino acids. Now, it has an amino group, NH2. Remember the last time that we talked about this? It's a functional group, one of the 7 functional groups. And this is called the carboxyl group. We know this because it has a C double bonded to an oxygen atom and then OH. So COOH is a carboxyl group. Now, in the center, there is a carbon binding to 4 domains. So we'll call it an alpha carbon, just to know that it is in the center, and we call it alpha carbon. And the other side is a hydrogen atom, and on the other side is a big, enormous group with numerous atoms, and collectively in this particular position, they are called the side chain, or you may call it the R group because it's represented by the letter R. Right, so these part, so amino, alpha carbon, hydrogen, and carboxyl group, they are all the same in the 20 amino acids that we'll be talking about today. They are all the same. Now, what makes them different would be the R group. So the R group comprises of different atoms in each amino acids, which gives them different properties, structures, charge, and anything that we need to serve as guidelines when we are trying to group the amino acids. So today we'll be mainly talking about the R groups, how the R groups may be different, and therefore how we may group the amino acids. So we've been talking about the grouping process. Now there are 4 groups. We'll be beginning with a group called hydrophobic amino acid groups. Now, they're hydrophobic because the R groups will not react very vigorously with water molecules. Now, we know that water molecules has poles. It has 2 poles. So it's polar. That is due to the greater electronegativity in the oxygen atoms. So a water molecule is polar. Now, if we say something that does not seem to be reacting with water, that is polar, we'll call it nonpolar, right? So these amino acids, these 9 amino acids, have their R groups that are nonpolar. So nonpolar side chains or nonpolar R groups. Right, so they are called glycine, that's an H, alanine, CH3, valine, leucine, then isoleucine, then methionine, phenylalanine, so phenylalanine, this is alanine, and tryptophan and proline. So these are very complicated names, and if you just remember the names in a random order, it can be a huge deal, and it can take hours for you to try to memorize only these 9 amino acids, because you will always forget about the name or the properties that gives the attributions that we need to group them in this particular hydrophobic group. So here is something that you may use to memorize it. It's a sentence. So we start with glaciers in Alaska victoriously make isolated prowlers. Oh, sorry, I move it. So glaciers in Alaska victoriously make isolated prowlers lose their phobia. Phobia. Okay. Now, why is this sentence a very good provides assistance when we're trying to memorize? Because the capital letters all corresponds to the amino acids, and by the last word phobia, we know that they are restricting. They are resistant to water, so they are hydrophobic. So phobia here actually corresponds to the hydrophobic, the group attribution. So glacier has a G that corresponds with glycine, and then we will have the Alaska. So Alaska corresponds to alanine, victoriously, valine, make. So it's methionine. Then isolated. So iso corresponds to isoleucine. Prowlers, proline here. Then leucine, or lose is here. So they are pronounced very similarly. So leucine, and there. Now this part can be tricky because tryptophan is a real long word. So tryptophan, there. Now the P in the word phobia has two overlapped meanings. One is hydrophobic, and the other is an amino acid called phenylalanine. Now, I would suggest that you remember it when you are trying to memorize alanine, because it's just like adding a prefix to the word alanine. So phenylalanine in alanine, and the same with leucine and isoleucine, because they are like very similar in the word, just the difference in the prefix. All right, so let's just do it again. Then we move on to another group. Right. So glaciers in Alaska victoriously make isolated prowlers lose their phobia. We know that they are in the hydrophobic group, and there are glycine, alanine, and we know the prefix. If we add a prefix to alanine, it becomes phenylalanine. Then victoriously corresponds to valine, methionine, isoleucine, proline, leucine, and tryptophan. Right. One thing I would like to mention here about methionine or MET is that when we will be discussing about the central dogma of genetics, we will know that a codon, the start codon, which is the particular place when the cell is trying to decode the RNA chain. So you start with a codon called AUG. You always start with AUG, and the AUG codon actually corresponds to MET methionine. So methionine corresponds to the start codon and vice versa, and so you may discover that the amino acid methionine is actually the place where you will have the polypeptide chain where it starts when the cell is trying to decode the genetic materials or genetic information. All right, let us move on to another group. Now, we know that this group is hydrophobic. We will ask ourselves, are there any groups that are hydrophilic, and they are. So there are only 6 amino acids in this group, hydrophilic. I'm sorry, hydrophilic. Now, hydrophilic is the exact opposite to the hydrophobic. Well, because hydrophobic is being resistant to water or non-reacting to water, then hydrophilic is water-loving, or it's attracted to, or it can stay within the same area or position or hold the same space with water molecules because they are hydrophilic. Now there are 6 called serine, threonine, cysteine, tyrosine, asparagine, and glutamine. So STCTAG. I'm just reading the first letters of this word, not the abbreviations or the proper names that are shortcuts for the amino acids. Now, like the part what we did with the hydrophobic group, like the glaciers, Alaska, blah blah blah, we also have a sentence particularly made for the hydrophilic group, and this is called 3-serine cyclist gluten-free asparagus. Now I know that this sounds silly because because asparagus cannot be gluten-free, and cyclists cannot really be serine, but this is only for you to memorize it. Okay, this is only for you to memorize it. So whichever way that you find interesting or helpful can be used. Now, let us see how this sentence corresponds to the amino acids. Right, so 3-TH, we have threonine, and serine. It sounds pretty much like serine in here. So 3-serine, threonine serine. Then cyclists, we have CYC, and we also have cysteine starting with CYS. So they are quite similar. And tri. So tri has TRY, and we also have an amino acid called tyrosine, and they start with TYR. So it's kind of like a self-reminder that TRY, TYR, tyrosine, or tri. And gluten-free, this is kind of straightforward because GLUT, gluten, corresponds to glutamine, and asparagus correspond to asparagine. So they are quite similar in pronunciation. Asparagine, asparagus. We have 3-serine cyclist tri-gluten-free asparagus, and corresponding to the amino acid, we will have threonine, serine, cysteine, tyrosine, glutamine, and asparagine. All right. So just trying to reiterate, I know this sounds a little bit silly, but you will need to try any approaches that suits you well and helps you with remembering these amino acids. Now, there are 6 amino acids that are hydrophilic, and one amino acid that I would like to spend some time talking about is tyrosine. Because tyrosine is widely used in cosmetics. Many cosmetics affect or inhibit the formation or the change or the reactions or the chemical reactions that tyrosine is involved. So that is because when tyrosine gets to change into other substances, or when it is involved in chemical reactions, tyrosine will eventually form melanin, which is basically black pigments. So many cosmetics, in order to make your skin more pale, they will impede the reactions in which tyrosine is involved. And another amino acid that we must pay attention to is cysteine. So Since cysteine is only weakly polar, it is sometimes classified as a non-polar amino acid. So cysteine is quite particular because SH and CH2, they're not very different in electronegativities. So it can sometimes be classified, in some books, it is classified as nonpolar, but in Campbell, it is polar with an annotation, of course. Now, SH, remember the last time that we talked about subhydral group that we earlier mentioned about the subhydral the subhydral group, and they can form disulfide bridges that actually holds the protein structure. Now, when we are forming the disulfide bridges, cysteine is the amino acid that is involved because it has the SH. Now, this also explains why the element sulfur is fundamental and essential for the process of life, because it is built into cysteine, an amino acid which can form disulfide bridges crucial to the structure of the proteins. Right, we have talked about 9 hydrophobic and 6 hydrophilic, that and therefore we will have 5 left to discuss about today. All right, so the 5, they are grouped into 2 groups. One is acidic, meaning that it's negatively charged, and the other is basic, meaning that it's positively charged. Okay, so these 5 collectively called electrically charged side chains, because they all have charges. They are all hydrophilic, because remember that the polarity of the water molecules, which makes some R groups hydrophilic and the other hydrophobic, is due to the electronegativity, which is the ability of attracting electrons around in one particular atom. Now, the electron will confer some charges to the atom. So it is, you may consider them the polarity as charges as well. So if we have R group that are electrically charged side chains, they are of course hydrophilic. Now, right, so we'll begin with basic because it is more complicated, and it has 3 components. One is lysine, the other is arginine, and the last one is histidine. So how do we remember this? It's quite simple. So basically, Argentine history is lovely. Now, basically means that the following amino acids are basic, just like what we did with the glacier part, the hydrophobic part, like phobia. Now, this part is basically. So these 3 are basic, and arginine corresponds to arginine. They are very, very similar in the word structures, argentine and arginine. In history, it has HIS as a starter, correspond to histidine, and actually it is abbreviated into HIS when we are trying to make shortcuts. So is lovely. Lovely, starting with an L and ends with a Y, corresponds to the amino acid lysine. So LY, L and Y. So basically, arginine history is lovely, corresponds to their basic, then arginine, histidine, and lysine. All right. So these 3 are basic. In acidic, honestly, I don't have any sentence for you to remember or to make the process of memorizing funnier, because come on, there are only 2 amino acids. So acids, they are all having the word acid built into their names, and one is aspartic acid, and the other is glutamic acid. So you may see that they are quite similar in the word structure to asparagine and glutamine. Okay, so the two that are acidic are negatively charged, and they are aspartic acid and glutamic acid. Right, so these are the 20 amino acids that you will need to memorize when we are talking about biology.